and hopefully it will record. Search Protein Lecture 1. And outline of this lecture is the dietary protein as a requirement. We'll talk about why we have to eat it. Then we'll talk about digestion of dietary protein, and then absorption, and then we're going to talk about amino acid metabolism, which I don't know how much you've talked about that in other classes. We're going to go kind of light on this. If you notice in the textbook, as you're reading this chapter is about 40-something pages. Okay, so in this chapter you can focus on what we're doing here because there's a lot of amino acid metabolism we won't talk about. I'll talk about some of what I think are the important points of this as we go through this, but we're going to condense this information. And one of the reasons we're covering protein like this is proteins should not really be an energy source. So we're not going to talk about the energy aspects of it as much as we are kind of the functional aspects of structural proteins and things like that. Okay, so with that said, why do we need to eat protein? Tastes good. Structure. Okay, so so it's important. Parts of it are important for muscle structure. Okay, that's a good point. Not one of my 2 points, but what do you think? Why do we have to eat protein?
To have amino acids available for building our own protein structures.
Okay, amino acids, more specifically, because they're a source of. Dietary protein is a source of essential amino acids. The amino acids that we either do not make or don't make in adequate amounts. What about any other reasons? What makes, protein, as far as the molecules or the elements involved, what makes protein different than carbohydrate and lipids? Nitrogen. So we eat dietary protein as a source of nitrogen. Nitrogen is one of the most common elements on Earth. A lot of that nitrogen is in its diatomic form. It's reduced and there's actually a triple bond between nitrogen atoms. We can't reduce that. We can't assimilate that nitrogen into our body. There's organisms that can. They're called nitrogen-fixing organisms, right? They can take that nitrogen and incorporate it in other molecules like amino acids. And then those amino acids become available to other organisms that consume those amino acids. So we consume the amino acids as a source of nitrogen rather than nitrogen itself. Okay, so we have to have it because of that. So keep that in mind as we go through this, because one of the things we can do to tell if a person's getting adequate amounts of protein is determine how much nitrogen they're excreting. And we can balance that with how much they're consuming. So we'll go through that. We'll talk about how do we know if a person's getting enough dietary protein. Okay, so those are the 2 reasons. So let me add a little bit to that. The second thing is it's a nitrogen source, right? That nitrogen can be used to synthesize non-essential amino acids. So we need it as a nitrogen source so that we can make non-essential amino acids and other nitrogen-containing molecules. So how many amino acids are used during translation to synthesize proteins? right? We talked about that a lot, right? mRNA, ribosomes. Ribosome is basically working with the mRNA to incorporate amino acids into a protein sequence. And how many amino acids are used during that process in us? 20. Remember that? We use 20 different amino acids. That genetic code for humans, 20 amino acids. How many of those amino acids are essential in the diet? 9. Now, it doesn't mean that those amino acids are not modified post-translationally, because some of them are. Right? We make a protein and then we make new amino acids that are part of that protein by adding a different chemical group. But during translation, there's 20 of them. Of those 20, 9 of them have to come from our diet. And a lot of those 9 are we can make. We just can't make adequate amounts of them. There's really only about 2 of them that we don't make at all. But we do need them in our diet because we don't make adequate amounts. Is there an RDA for protein? And what is that? Remember that number? Remember that number? The RDA for protein is, let's say for this group, adults. No, it might be about that, but we generally do it, what, grams per? Yeah, yep. For adult women, 0.8 grams protein per kilogram body weight. So it might end up to be 50 grams, but it's on body weight. How about for same age group, adult men, what is it? That is a trick question. It's the same. Okay, 0.8. Seems weird, huh? Yeah, and it probably needs to be adjusted. Okay. What type of activity does that assume? Nothing. Sedentary. So the RDA is for sedentary individuals, healthy individuals, and those individuals of healthy body weight. We'll talk about that as we go through this. So there's an RDA for protein. Do you think there's an RDA for any of the amino acids? So the amino acids, you're right, there is. You're right. You're absolutely right. What do you think? Is there an RDA for any of the amino acids? It would make sense for the essentials. Is that the case? Yep. We've all thoroughly looked through that protein chapter. If you look towards the end of that protein chapter, you'll see a bar graph where it says, hey, these are the RDAs for the essential amino acids. You'll see individual amounts. And in that case, you can look at that and go, oh, some of these essential amino acids tend to be required in higher amounts than others. So what does that lend itself to? Wow, are some dietary proteins better at giving us the essential amino acids than others? Absolutely. Have you ever heard people say things like, oh, that's a complete protein, or that's a high quality protein? We're going to talk about that next time. That's why you say that, because you look at the relative abundance of essential amino acids in a food relative to our essential amino acid needs. And you can actually do calculations based on that. So keep in mind as we go through this, these amino acids are present in different amounts in different diets. And the quality of those diets are different. So the RDA is kind of a ballpark number, and it's assuming where you're eating a mixed diet and you're not exercising. As soon as a person exercises, protein requirements go up. And I think everyone in the nutrition world has let go of no, they don't. But I remember when I was an undergrad, the person I had these classes from said, oh, no, you don't. I'm like, I think you do. But now it's kind of irrefutable. It's like absolutely. So I'll give you numbers on those as we go through this. So that's where we're going. So we got to make sure that our dietary protein gives us the adequate amounts of these guys. These are the essential amino acids for humans. You can also refer to it as 9 nutritionally indispensable. In other words, they have to be in your diet. You got to memorize these. Do you know these already? Okay. You got to know which ones are essential, which are not. So these are the 9 essentials. And if you want to, you can use like a mnemonic device. You can go Private Tim Hall. Remember that one? Apparently not. You don't remember Private Tim Hall. So PVT, you can use the initial on the front, the first letter of each of these and make it accurate however you want to do that. But you got to remember these. Okay? Let me tell you a few unique aspects of some of these. The top 3, leucine, isoleucine, valine. Have you heard of those? Kind of famous. Why? Those are the branch chain amino acids. Why they call them that? Because their chemical structure has kind of a branch point in its side chain. Leucine, isoleucine, valine. Those are the branch chain amino acids. Of the 20 amino acids that are used for translation, only 3 of them are branch chains, and they're all essential. And they're commonly used for proteins in our body. Of those, probably the most interesting one to me is leucine. Leucine's also a signaling molecule. Have you guys taken like classes in biology classes where they talk about the trip operon and how tryptophan actually signals transcription and things like that? For us, leucine tends to be that molecule. Leucine is an important, I'd say, anabolic signaling molecule in humans. Its signaling pathway, kind of parts of it parallel the insulin pathway. Some of it is very unique, but it signals for protein synthesis and anabolic metabolism. Because what the cells are doing is they're saying, well, if there's adequate amounts of leucine, it's probably a high quality protein source, so we can make proteins. If leucine's there, there's probably other amino acids, so it signals protein synthesis. The other ones that we're going to talk about, and do you know the structures of these? Any of you? I think we should, but we don't have to. I'll only give you 3 of these that I'll expect you to know the structure for, and we can make some points with that as we go through this. So remember, here's an important part. The genetic code determines the protein's amino acid sequence. We've said that several times. So what if the genetic code specifies for an essential amino acid? Let's say this mRNA is combined with a ribosome and translation elongation is occurring. And now this ribosome comes along and it comes up to a codon that specifies, oh, we need leucine because of this codon. If that tRNA in the cytosol is not adequate, loaded adequately with leucine, in other words, if leucine is compromised, then that tRNA won't go into the ribosome to continue elongation. So what happens to protein synthesis? Stops. So it's an all or none process. So if a person is not consuming adequate amounts of dietary protein, it can affect protein synthesis. And that's one of the reasons it becomes lethal, if you're not getting adequate amounts of protein. There's 2 main places that cell can obtain amino acids. One of them is what? Skeletal muscle. So in other words, we can, if the person is in catabolic phase, then some amino acids are going to be generated from proteins that are being degraded. And then those amino acids, some of them can be incorporated into proteins. The other source, the diet. So it's important that we consume appropriate amino acids to support protein synthesis and metabolism without having to catabolize any of our own proteins, because we really don't store proteins in a storage form. They have a functional role Okay, so that's what happens. And you, I'm sure you guys have seen pictures of individuals that are malnourished or starving, and you see basically the way their physiology and their kind of wasting of tissues. Most of it is kind of a random catabolic process where all different types of proteins are being broken down. And it's trying to, in that case, a lot of times it's to provide an energy source and a glucose source. And so proteins are broken down and they can't be resynthesized. So if you go on Survivor, keep in mind, probably one of the first things that you want is water. It's the most important nutrient short term. And then you could probably argue, well, probably the next one is some water-soluble vitamins. So keep that in mind for next quarter. And then one of the top ones, protein. Got to have a nitrogen source. Okay, and we'll talk about that coming up. Remember that if you're on Survivor season 28 or whatever it is by now. All right, so remember the structure of amino acids. All amino acids have the same core structure. And we're just talking about the 20 that are used for protein translation. Remember there's some other ones like taurine and some other amino acids that aren't used for translation. But if we look at their structure, I'll review, I know. They have a central carbon, known as the alpha carbon. They have an amine group, carboxylic acid group, and a side chain, sometimes referred to as the R group. So if you look at glycine, glycine is famous because it's the simplest amino acid. Let's remember that one. One of the ones that you're going to remember the structure of is glycine. Okay? That's one of 3. The other 2 amino acids that I want you to know the structure of is phenylalanine and tyrosine. And you'll see why I picked those 2. Here's phenylalanine. All right, central carbon. So all you have to do is off your central carbon, CH2, aromatic ring. That's it. What's the difference between phenylalanine and tyrosine? Hydroxyl group. Tyrosine has an OH right here. So that's not too bad, right? So you need to know be able to draw the structures of glycine, phenylalanine, and tyrosine alone and in a peptide. And then that'll be in the study guide. At pH 7. Okay, so that's what makes them unique, that side chain. Uh-huh.
So at pH 7, will the OH have the H or will it be negative?
It'd be like this. Hydrogen dissociates. So here's one amino acid. And if you put an H right here, that's glycine. Here's the amine group. It's positively charged. So when you draw this, remember your nitrogen, remember formal charges and all that. If nitrogen has 4 bonds, that nitrogen has a positive charge. So this is H3N. Central carbon. This is always a hydrogen on that central carbon. Here's your carboxylic acid group. It's going to look like this. Okay, so you'll draw them individually and then you'll combine them. When you combine them, water comes off. So that peptide bond is formed between the carboxylic acid group of one amino acid and the amine group of the next. So when you're making a protein, this is what those ribosomes are doing. They've got an enzyme that basically make that peptide bond. So water's generated. So there's your peptide bond. And you guys probably talked a lot about that in biochem or something, the structure of that, how it's planar and all that. Okay? So you got to remember that one. And a shortcut to this, I don't know if you remember this, but if you draw a protein or a peptide, your backbone is always NCC. NCC. So if I have another one here, it's going to be NCC. So you can just add them. So remember, your first carbon, that's your central carbon, so you're going to put your side chain here. Your carboxylic acid group is shown here. It's now just a double bond oxygen, and it's bound to the nitrogen. And your nitrogen always has a hydrogen that's basically opposed on the other side like that. It's typically the way you draw it. Okay, that's a covalent bond. So digestive enzymes and proteases that break that peptide bond a lot of times use water as the electron source. They'll hold on to a water molecule and add it to that peptide bond. So you should be able to draw the ones I mentioned, then identify the amino and carboxy terminus, right? We know this is the amino terminus, carboxy terminus here. Good, and don't forget the water when you combine them. I'll say account for all atoms, so you've got to show that the water comes off. Okay, so just a brief overview. I'll tell you why we're going to look at this. This is out of a different text. Physiological roles for protein and amino acids. So the left side is protein, right side is amino acids. You've talked about this in a lot of different classes. Proteins function as enzymes, antibodies, hormones. They're involved in pH regulation in both intracellular, extracellular. They regulate fluid. So for example, if a person is compromised in protein intake and they make less albumin, that albumin is no longer able to hold water within the blood vessels. So the water starts to leak and you end up with things like edema in the extremities, but also sometimes abdominal edema, like in kwashiorkor and some of those malnourished states. And that's what's happening. So sometimes you can look at a blood test and measure a person's albumin and determine their protein status. If they're malnourished, albumin tends to stay in the blood about 14 to 18 days. So if they're not eating adequate amounts of protein over periods of weeks, albumin tends to drop. So sometimes that's an indicator. It's not real sensitive, but sometimes it's part of the process of piecing together their nutritional status. Blood clotting, you'll talk about again in 329, cellular blood transport like lipoproteins, and then movement, skeletal muscle. We're going to focus more on this. This is amino acid metabolism. Now, some amino acids are used to make other molecules because they have nitrogen. Or sometimes they're used to make proteins. So they're used to make nitrogen-containing molecules. We'll talk about some of those today. Some of these amino acids are actually used. They're oxidized. So in that case, they're used as an energy source. Remember we looked at the diagram last time, we saw those amino acids going into Krebs cycle. In that context, we said, well then oxaloacetate can basically become glucose. But sometimes those amino acids are just being used for energy. And typically it's not a lot, but it does happen. So we lose some amino acids as a source of energy. And then a lot of it's used for make glucose. Almost all the amino acids are gluconeogenic. Almost all amino acids can become glucose. So we're going to talk about some of this. So a little bit more background. How much protein is in the body? Well, protein is about 14 to 17% of the total mass of a lean adult. Now that's actual protein. That doesn't mean lean body mass. A person has a lot more lean body mass than that. That's the actual protein. In other words, the water's removed everything. That's just protein. So for example, about 28 to 30 pounds of a 200-pound man would be protein. If you look at the overall kind of protein categories in the body, skeletal muscle accounts for about 40 to 60% of body protein. That's where most of it is. Most of it's in skeletal muscle. So for example, if I scan a person and I have their body composition in front of me and I see a number that says this is how much lean body mass they have, you can guess about 70% of that skeletal muscle. So you can see if they're changing lean body mass, there's a good chance they're changing the amount of skeletal muscle. Skin and blood protein account for about 15% each. So about 15% of the body's protein is in the skin. about 15% of the body's protein is in the blood. And that would be things like albumin, antibodies, some of the other more common proteins, hemoglobin, all that. So let me give you a couple points about this. You know about approximately how many proteins are estimated to be in the body? 1,000, 2,000. Remember the Human Genome Project? Fantastic, right? Changed your life. What do you think? One of the numbers they came up with is they said, wow, we now have this genome sequence. They've said, now let's look for open reading frames. Let's look for potential sequences that generate proteins. Have you ever heard that estimate? It's estimated that there's probably about 30,000 different proteins in our body. 30,000. I'd probably say most of them, we don't know what they do. But it's estimated that they're there. We're still figuring ourselves out. But if you look and you said, well, which are the most common proteins? There's 4 of them that tend to be more common than any of them, and I'm going to tell you those. Here's the 4 most common proteins in the body. As a matter of fact, about half of the body's protein is contained in just 4 different proteins. First one is collagen. Have you heard of collagen? Well, what does collagen do? What's that? Yeah, it's part of connective tissue. So it's the most important protein in the extracellular matrix. Basically, it holds cells together and tissues together. So collagen's a structural protein. There's 2 other very common structural proteins, and I bet you could guess them. Actin and myosin. So those are the structural proteins: collagen, actin, myosin, and the oxygen transporting protein, hemoglobin. Those are the 4 most common proteins in the body. Now think about this. If a person's malnourished or they need glucose or they're trying to, do anything like that to obtain energy, you can imagine the proteins that are most susceptible to degradation is going to be that actin myosin, proteins within skeletal muscle, because that's where most protein is, and those are the most common proteins there. So in that case, you're actually breaking down proteins. So here's kind of another interesting way of thinking of this, or looking at this, at least for me anyway. There's 20 amino acids in the body, right, that we use for protein translation. But they're not equally distributed in proteins. In other words, some of those 20 are used a lot more commonly in proteins than others. But here's an interesting point. 2 of the most common amino acids in the body's protein are essential amino acids. 2 of the most common amino acids in the body's proteins are essential amino acids. And that is leucine and lysine. Sorry, those are leucine and lysine. So if we don't get those in the diet, it's going to affect a lot of the proteins and their metabolism. Now let's talk about this one. This one's pretty interesting, I think. Have you ever heard of protein turnover? That phrase? How about this one? Use it or lose it. Right? So protein turnover is the dynamic balance between protein breakdown and protein synthesis. In other words, proteins in our body are constantly being turned over. Some proteins are being made while other proteins are being degraded. And it is a significant process that's going on 24/7. And if you look at this little diagram, you can see that there's a couple important points here. Let's say this is a protein in the body. That protein can be degraded. So the amino acids are available. Those amino acids can be reincorporated or reutilized for another protein. Or they can be catabolized. In other words, if we look back at their structure, let's say this protein comes out of this amino acid comes out of a protein. This amino acid could be degraded itself. In other words, the nitrogen can be split, and now you have a carbon skeleton. That's what that's called. And that can be used for other things. It's no longer an amino acid. So in that case, you've pulled an amino acid from a protein, and it's no longer used for protein synthesis. So one of the things to keep in mind is kind of how common this is. And which of these is more common? Let's say we have a protein that's degraded. Which of these do you think is more common? Reutilization or catabolism? Reutilization. I seem to ask questions that you have no information to answer, so I'm just asking for you to guess, right? That's not very good. And here's the reason that's so important to think about this. There's a number, there's a way that we can estimate how much protein is undergoing protein turnover in the body at any given time. So in a 24-hour period, we can estimate how much protein in the body has undergone turnover. And here's the way you calculate this. That number tends to be about 4.6 grams of protein per kilogram body weight. So let's say a person weighs 70 kilograms. Is that a lot? What do you think? So what this is saying is, in a 70-kilogram person, and we're just ballpark, in a 24-hour period, approximately 320 grams of protein in their body has been degraded and gone through this process where the amino acids are either reutilized or oxidized and used to make other molecules. 320 grams of protein in our body in a day. How many of you guys eat about 320 grams of protein a day? Because really that's what's happening here. If we say, oh, it's turned over, in other words, this protein has gone from protein to amino acids. 320 grams of it. If all of those amino acids went into catabolism, we would essentially have to eat 320 grams of protein to replace those amino acids. How many guys eat 320 grams of protein a day? Not many. Approximately how many do we eat? 45, maybe up to 100 grams of protein, something like that. Way less than that. Why can we eat a lot less protein than that number? Because we reutilize amino acids. That reutilization of amino acids tends to be the most significant amino acid source for protein synthesis. So the most important source of amino acids for protein synthesis are endogenous amino acids. The most important amino acid source for protein synthesis is endogenous amino acids. Okay. In other words, if, for example, let's say there's an amino acid that's part of a structural protein in my forearm and it's degraded, it goes into the blood. So on Monday, maybe that amino acid is functioning in myosin, and on Tuesday, it's part of a hepatic enzyme. And it could continue to do that. But keep this in mind. That flux in protein metabolism means that in order to retain those proteins, we need to be in anabolic phase for the appropriate amounts. In other words, as soon as we get sick or as soon as we become bedridden, more of those proteins are lost. And that's when protein metabolism becomes completely different. So there's times where we want anabolic phase, there's times where we want catabolic, but in general, we want a balance. We want amino acids available, so we need proteins to be broken down, but we also need to have an anabolic profile. In other words, especially hormones like insulin, to be available at the appropriate time so we can reincorporate those amino acids. If not, we lose too much protein and we become a nitrogen deficit, and that's when our health can be really compromised. As soon as a person becomes bedridden, for example, like stress, they lose a lot of protein because all this protein that's turning over, now they're having to use some of it for things other than making proteins. And that's why they can lose so much skeletal muscle so quickly. All right? And as we get older, of course, and other things that happen, if a person's cachectic, if they have cancer, if they have anything that's significant, that can significantly affect protein metabolism and how those amino acids are used when they become available. So we have to keep stuff like that in mind.
How does that affect people with PKU?
We'll go through that. Good question. Yep. Good there? Yep. They yeah, it's difficult to say all the amino acids have kind of a set half-life because the conditions affect that. Yep. So good question, but it kind of depends on the conditions where that amino acid is in the body as well. Yep. Because some proteins only stay around for a few minutes while some other ones stay around for weeks. Okay, let's talk about digestion absorption. And again, we'll go through some details here, and keep in mind we're learning more details than we did in the digestive system, and I'll tell you why as we go through this. So digestion of a protein involves denaturation by hydrochloric acid. In other words, denaturation is different than digestion. So this is a protein that has, let's say it's native conformation, it's got its normal structure. Hydrochloric acid or heat changes the interactions between the side groups, and now the protein changes shape. It's now denatured. The denaturation of the enzyme of the protein is important because now it makes more of those peptide bonds accessible to digestive enzymes. Right? So imagine, let's say we have a protein that's all kind of in a tight molecular structure. The enzymes can't penetrate certain parts of the protein and can't access the peptide bonds. So it's important that it's linearized. Two ways to do that. And that's heat and hydrochloric acid. So that's what that means. So just a quick review, right? Remember, protein digestion starts in the stomach. As soon as we consume protein, then gastrin's released, stimulated by presence in the stomach, and this stimulates the release of hydrochloric acid from parietal cells. The hydrochloric acid denatures the food protein and also activates pepsin from pepsinogen. And then pepsin is that endopeptidase that basically starts breaking down, digesting proteins in the stomach. So protein digestion starts in the stomach, but it's nowhere near complete. But protein denaturation should be complete in the stomach. And now the partially digested proteins and denatured proteins enter the small intestine. So this looks a little messy. It's actually kind of simpler than it looks. Let's imagine stomach's here. So now these proteins are coming into the small intestine. So this is the small intestine. Remember, the small intestine is lined with enterocytes, major function, absorption. There are also some endocrine cells. that are responding to the presence of molecules in here. And for example, some of those endocrine cells in the small intestine release CCK. And CCK affects the pancreas. Remember we said CCK is the major effector of pancreatic exocrine function. So once CCK hits the blood, the pancreas releases enzymes for protein digestion. Now, this is an example of a process we talked about before. These are zymogens. These are inactive enzymes. They come from the pancreas, and then they're activated in the lumen of the small intestine. All of these are inactive enzymes. The enzyme that starts their activation is enteropeptidase. So enteropeptidase is attached to enterocytes. It's kind of always there. So enteropeptidase is attached to enterocytes. Enteropeptidase converts trypsinogen to trypsin. Remember trypsin? When did we talk about that? Isomaltase and sucrase. We said trypsin comes from the pancreas and cleaves that protein. Trypsin also cleaves a lot of other proteins, including these other enzymes. So enteropeptidase activates trypsin, which activates additional zymogens, like chymotrypsinogen, proelastase, procarboxypeptidase A and B. And then look what happens to them. They become the active enzymes, chymotrypsin, elastase, and carboxypeptidase A and B. Now, so all of these are proteases. So what's their target now? Dietary protein. So why do you have so many of these? What are they looking for? What are they looking for in a protein? How do they know where to cleave? Amino acid sequences. They're going to look for a specific amino acid sequence or a type of amino acid and cleave right next to it. So keep in mind, every protein has 20 different options at every amino acid site. So there's unbelievable combinations of amino acids that make up all these proteins that we eat and they're in our body. So what that means is you need a lot of different enzymes to recognize all those different sequences. So there's a ton of these. This is a very short list. So these are active proteases that are in the lumen of the small intestine. Some of them are exopeptidases. Some of them are endopeptidases. Remember the difference of that? Before, when we talked about pepsin, we said pepsin is an endopeptidase. Of these enzymes, the endopeptidases, well, actually, let's do that more easy for you. The only exopeptidases are these guys, carboxypeptidase A and B. These are the only exopeptidases. In other words, they're taking the amino acid off the end. And which end are they working on? The carboxyl terminus. Why is there an A and a B? They have slightly different preferences. One of them will go, oh, I like hydrophobic amino acids, I'll cleave that one. The other one will say, hydrophilic, I can cleave that. So these are the only exopeptidases. The other ones, elastase, chymotrypsin, trypsin, enteropeptidase, endopeptidases. So all the other enzymes are endopeptidases. They're looking for internal peptide bonds. Now, so the result of this is you get some free amino acids, and you get some peptides. Some peptides are cleaved by more enzymes on the brush border membrane. So it's kind of like carbohydrates, where you have amylase and then the saccharidases that are on the surface of the enterocytes. For protein, it's the same thing, kind of two phases. Got the brush border membrane is kind of in phase 2. And there's 2 categories of peptidases on the brush border membrane. There's aminopeptidases, which are cleaving the amino terminus amino acid, and then carboxypeptidases, which remove the carboxy terminus amino acid. So the products of brush border membrane peptidases are free amino acids and a shorter protein. Let's do one more slide. This one's pretty quick. Here's a difference, and I mentioned this before when we talked about digestive systems, so remember this. This is different than carbohydrates. Remember, carbohydrates are converted to monosaccharides. Proteins are absorbed mostly as peptides, not single amino acids. When you eat protein, we go home tonight, we have a dietary source of protein. That protein is going to go through the stomach. It's going to be partially hydrolyzed. It's going to be denatured, goes into the small intestine. 33% of that protein is going to be absorbed as single amino acids. And there's lots of transporters. I'm not going to drive you nuts with all this. There's a lot of amino acid transporters that have slightly different characteristics. And that's this carrier. That's probably a transporter. Okay. Absorption into cells require transporters, and there are multiple types. Many use sodium in a similar way to the SGLTs. Remember that? Sodium binds, affinity for the amino acid or the substrate increases, transported in. Is that energy dependent? Yep, and it's based on that sodium gradient. So does some absorption of protein require energy? Absolutely, because it requires sodium. Okay, so there's lots of these. There's also a peptide transporter. And I think there's one that does almost all peptides. It's called peptide transporter 1. And it moves di and tripeptides into the intestinal cells. Most protein is absorbed as di and tripeptides. 67% of it is. So that dietary protein from the meal, most of it is going to be into enterocytes as di and tripeptides. How about whole proteins? Do we absorb those? Very little. What's one of the reasons that's important, that we don't absorb whole proteins or big pieces of proteins? What could that cause?
Leaky gut.
What's that?
Leaky gut.
It can be from leaky gut that allows that to happen. That's what we call leaky gut, is when those gaps in between the enterocytes are not tight enough and molecules go in. That can cause an immune response. It can stimulate allergic reactions, things like that. Proteins are the ones that are going to do that. So it's important that we break down those proteins so that we don't stimulate the immune system. But peptides is what goes in. Good there? Okay, guys, if it's okay with you, I'm going to stop there. Any questions? Okay. So I'll stop there, and I'll send you an email as soon as I get the assignment done. But remember, next time is when the extra credits due. Thank you.